now students, we are taking up some more examples, and if we have taken the coelant traits into account, then after coelant traits we have got a phylum which is of parasites, and this is of Platyhelminthes. Now if we take this particular phylum into account, we have got Protonephridia, which are having flame cells over them, and these are the excretory cells which are present in the Platyhelminthes. So these are the representative kind of animals which are showing the flame cells as the excretory cells. Now these are collecting excess of the water in addition to the nitrogenous wastes, and they are emptying into the nephridiopore, and they are excreting the ammonia out of the body of the animal. Now if we talk about these animals, so here we have a protonephridium, and it is a network of the tubules which have got dead ends. Now why they are having dead ends? Because they are having flame cells which are closed on the top. As a flame cell has got a number of cilia which are vibrating uh, on both the sides, and they look like as a flame. That's why they have been given the name of flame cells, because of the presence of the vibrating cilia. So they are lacking any kind of internal openings. So they are blind protonephridia which are present in Platyhelminthes. And if we go with the structure of a typical Platyhelminthes, this is Dugesia. This particular animal is Planaria or Dugesia. So here you can find out the protonephridia which are present in whole of the body of this animal, and these are present on both the lateral sides of the animal. And now what they are doing is that the flame cells, this is a single flame cell, or you can here see this flame bulb, and this is again a flame cell. And these flame cells, if we see the internal structure of these cells, they are containing cilia, and these cilia, when they move inside the lumen of the flame cell, the pressure decreases inside the flame cell. So this decreased pressure, it exerts a pulling pressure on the, you can say, the excretory materials which are present outside the flame cell. So when the cilia move, the pressure decreases inside, and the excretory materials from outside, it is coming towards the lumen of the flame cell. So ultimately, with the movement of the cilia, all the excretory products which are present outside the flame cell, they are coming towards the lumen. So ultimately, all the things, they come into the lumen of the flame cell. This is called as the ultrafiltration process. So this is the first step to form the urine in case of Platyhelminthes. So these are all the things which we have done in BSc, but I am just repeating those things very briefly. So the excretory products which are now present in the lumen, now they are called as the primary urine. So this primary urine is formed inside the flame cell due to the process of ultrafiltration. Now this ultrafiltration process has occurred because of the movement of the cilia. So when the cilia they move, the pressure decreases, ultrafiltration occurs. Now the excretory products, they are in the lumen. Now if there are some important things which have come into the lumen, they will be taken back, and that will be taken back from this particular part, which is the capillary tube. So if you see a flame cell, and this part is the capillary tube which is below every flame cell. So every flame cell has got a capillary tube, and this capillary tube is very thin. This is a single celled thin tube from which the important things which are required by this animal will be taken back, and those will be coming from the capillary to outside. So this process is called as reabsorption. So as we have discussed the excretory structures in the prior animals, so here it is a very equivalent kind of a structure to our nephron. So in ourselves, in humans, we have these two processes, the ultrafiltration and the reabsorption, and the same two processes are present over here in a protonephridium of Platyhelminthes phylum. So that was the physiology of the flame cells. So ultimately, after the reabsorption, what we have inside the capillary, these are the only and only the waste products which are going downwards from this capillary section. So those will be coming down and those will be reaching this particular part which is called as the nephridiopore, and this nephridiopore is opening onto the body wall. So that means everything which is coming down, it will be excreted out of the body of this animal. So this particular pore will excrete out the things out of the body. So this is the process of excretion. So along with this excretory products, water will be given out of the body, and that is osmoregulation. So I always tell students that there is a very big role of excretion with osmoregulation. So that's why these two systems, they are all together, they are same, and they are performing like an equilibrium kind of function. So they are maintaining the homeostasis condition inside the body of the animal as far as the ions and the water are concerned. So students, that was about the excretory procedure of these flame cells. So what they do is that the tubules, they excrete a dilute kind of a fluid. So they will be taking the fluid along with the excretory waste products that will be coming inside the lumen, and that fluid will be going out in the capillary, and from this capillary it will be going out of the body. So they produce a dilute kind of a urine for this particular animal. So these animals, as they are living in fresh water conditions, or they may be some may be parasites also. So depending upon the conditions, they may be giving a dilute urine or a more dilute urine or a very high dilute urine. So it depends upon the conditions in which particular platyhelminth is living. Now after this, students, we have the phylum which is Annelida. Now annelids, they are having metameric segmentation. They have got a particular coelomic cavity inside each and every segment, and each segment is separated from inside also. Now these plate-like things which are separating the segments from inside, they are called as septae. Single is called as septum. So these septa which are present inside these annelids, they are separating the coelom from each other as per the segments are concerned. So each segment has got its own coelom and own coelomic fluid. So you can say this is a metameric segmentation. So the kind of nephridia which are present in these annelids, these are not protonephridia as we have discussed in Platyhelminthes group, but here we have the metanephridia type. Now first of all, I should explain to you that what is a protonephridium and what are metanephridia. See, protonephridium has got a flame cell, or it has got a solenocyte. Flame cells were having many cilia. A solenocyte is having a single flagellum inside it, And if you have a flame cell or a solenocyte over a nephridium, it is a protonephridium. If we talk about the metanephridia, which are present here, metanephridia are not having any kind of flame cell or solenocyte, but they are having a funnel-like structure which is forming their mouth, and that is called as a nephrostome. So these are the opened nephridia, these are open nephridia, whereas the protonephridia are the dead end nephridia. They are closed, they are not opening out. So that is the major structural difference between the proto and the metanephridia. And if we go by the evolutionary process or evolutionary trend, you can say, in animals, the protonephridia were seen in the earlier phases of the animals, whereas the the metanephridia, they have developed in the later stages. So in the more, you can say, evolutionary higher animals, you may find the metanephridia. In the lower animals, you are finding the protonephridia. Now, in annelid, if we take the example of, suppose I'm taking example of metanephridia in earthworm, suppose. So here, what are metanephridia? They consist of tubules, and what they collect is, they collect excretory products from the coelomic fluid. So as we have discussed, in case of annelids, you have the metameric segmentation. So each and every segment is separate. They have their own coelomic fluid, and the type of nephridia are the metanephridia, and these are excreting out the waste products in different manners. Now, the type of the excretory product is the same for all the nephridia, but the type or the mechanism of excretion is different for these nephridia. So I'll be taking up the mechanisms one by one. And what they are producing is that they produce dilute urine again. So here, these metanephridia, they are again producing dilute urine. So these animals, they have to go for the conservation of the salts. They need salts, so they conserve salts, but they give out the excess of the water. So that's why they are giving out the dilute urine, and they are retaining the salts. So this is osmoregulation. If I take this particular structure, you can very well see this is a typical annelid metanephridium. Now, if we take earthworm, say for example, these are the segments which are altogether different or separate from each other. And here in the center we have the septum. These are the septa which are present inside the segments. Now, this is a particular metanephridium, and it has got a mouth which is called as a nephrostome. That's why it is metanephridium, because it is having a mouth, and this is opening in the coelomic fluid. As you can very well see over here, the mouths are opening into the coelomic fluid, so they are bathing in the coelomic fluid. So you can see like this. So if you have like excess of excretory products in the coelomic fluid, so these metanephridia, they are present over there, and they are taking in the excretory products like this. From the coelomic fluid, these products will be coming into the body of the metanephridia through these nephrostomes. Now, due to the, you can say, the excess of these excretory products, these products first of all enter the mouth, then they go into the tubule, and in this tubule the salt concentration is maintained. So if we go by the single structure of this particular nephridium, you have gone through these structures in your BSc. So in Earthworm, you have got three types of nephridia. I'm not going about the types here in this particular lecture, but if you go with the types of these metanephridia in annelids, we can have three types of nephridia. Number one, they can be pharyngeal nephridia, which are pouring their excretory products into the pharynx. Second type is the integumentary nephridia. So they are present all over the body and they are excreting out simply on the general body surface of the earthworm. So that is the second type, the integumentary nephridia. If we talk about the third type, it is the septal nephridia, and they are present on these septa and they are numerous in number on a single septum. So in BSc we have gone through the arrangement of the septal nephridia also onto a single septum. So what they do is that they also pour their secretions into the intestine. So ultimately the secretions, they are going out either through general body surface or through the intestine via anus. Along with this excretion, the coelomic fluid, it is also going out. So to secrete some of the salts, dilute urine is given out by the outpours of these metanephridia. So salts are conserved, dilute urine is passed in this case. Now students, if we go with the insecta, or you can say the arthropods, here we have many examples. Now here we have the Malphigen tubules, which are working 24 hours for the insects. Now these Malphigen tubules, they are present on the hindgut of these insects. This is a very you can say stochastic kind of a thing, that Malphigen tubules, they structurally they are a part of digestive system, but they function as excretory structures. So this is a very big kind of ambiguity. They are a part of digestive system, but they are working for excretion. So what they do is that they are taking the nitrogenous waste products from the body and they are excreting out the urates or the uric acid. So this is the main function of the Malphigen tubules in insects. Now here, from where they are collecting these nitrogenous waste products, they are taking it from the hemocoel. So here in the insects, we call blood as the hemolymph. So they are taking all the excretory products from the hemolymph and they are excreting uric acid. Now this particular structure is showing you the functioning of the Malphigen tubules. So you can take these diagrams from anywhere. You can take from Google, you can go with some encyclopedia, you can have Britannica, you can have Encarta encyclopedia, you can have some physiology book of like any author. So that can give you these diagrams. Now these small tubules, which are yellow in color over here, they are the Malphigen tubules. And these tubules, they are present on the hindgut of the insects. Now here you can see the Malphigen tubules. Now there is one thing which is very important to note over here, that salt, water and nitrogenous waste products, they are going inside these tubes. And what these tubes they do, they concentrate the salts, they concentrate the nitrogenous waste products in them, and then they excrete these products into this hindgut. So ultimately these products will be going out through the anus. So this is a kind of mechanism of excretion in case of insects. So what they are doing for osmoregulation, that they are collecting from hemolymph. It can be excess of water, but in these cases, the water is conserved. So as the insects, they require more of the water as they are not drinking much of the water and they are producing it metabolically. So what we call it as the metabolic water. So they are for the conservation of the water. And these tubules, what they are doing is that they are concentrating the salts, they concentrate the urates, they concentrate the uric acid, and then that concentrated uric acid is thrown out into the hind gut. So they excrete it out. So they are not using excess of water to excrete the uric acid. So students, that was the case of insects for the osmoregulation. Now, if we go with some environmental conditions, so what we have is that the animals that straddle multiple environments, they must have flexible homeostatic mechanisms. So this is very important thing, flexibility. So as we have discussed earlier, there can be three conditions which give you flexibility for homeostatic mechanisms. So what are homeostatic mechanisms is that if you require salts, what will be the mechanism? That you give out dilute urine. That means less of the salts will be given out. And if you are not requiring salts and you have to give out those salts as you are living in the marine water, then you will be giving concentrated urine and you will be conserving water. Now suppose if a particular animal is terrestrial and it is living on land, suppose, then it will be requiring water. So the requirement of that particular animal will be to conserve water. As we do, we need water. So what we do is that we drink water, and we have been prescribed that you have to take 5 liters of water daily. Now, why is it so? Because to remove the excess metabolic waste products, we require water. So, if we are not drinking water, what will be happening? That we will be giving out concentrated yellow colored urine. So, if you are drinking, moderate kind of moderate amounts of water, then your urine will be light yellowish. And if you are taking excess of water at a particular time in a day, you can give out a pure kind of, you can say, transparent kind of urine. So that will be having very less salt concentration. So this is called as the flexibility in the homeostatic mechanisms to cope up with the variable ion and the water levels. So this is very important, and each and every animal has its own efficiency to cope up with salt and water. Now students, here in the fishes, we are taking some strategies. So here we have ionoregulatory and osmoregulatory strategies of aquatic animals, and those can be distinguished by some of the factors. Now the differences between the extracellular fluids and the external conditions. This is a very important thing to note. Now what are extracellular fluids? These are the fluids which can be present in between the cells. These are outside the cells. Suppose in sponges, or you can say coelentrates, we have extracellular fluid circulation, or you can have the intercellular fluid circulation. And what are the external conditions? The amount of the water, or you can say the concentration of the water which is present outside the animal. So these will give you a particular strategy that how you will go for the osmoregulatory process. Now the second thing is the extent to which the extracellular fluids change when the external conditions change. Now this is a very, very important thing. Now students, suppose the outside water is now going lower in salt concentration. Now it is at 25. After 1 minute it will be 20. Now what the animal will do, animal has two conditions or two fates in this particular reduction of the salt level change. Now if the outside water is getting lower sodium ions in 1 minute, what the animal will do, number one, it can regulate its internal body fluids and it can regulate a 25 point sodium ion concentration inside the body. Number two, what the animal can do, it can change its internal fluid concentration to 20, which is very comparable to, which is matching to the external environment. So these are the two things which an animal can do in this particular condition. So what I am trying to say here is, this is the extent to which extracellular fluids can change when the external condition changes. Suppose if you have, I'm giving you another example. Suppose if particular animal is living at 25. I'm just giving you an arbitrary number. If any animal is living at 25 millimoles of salt of sodium in a sea in a very good way, then if we reduce the amount of the salt from 25 to 20, okay, what will happen? This animal will go for 20. Okay, no problem. The internal environments of the animal will change from 25 to 20 and they will be very equal to the external environment. If the external environment further changes from 20 to 15, what the animal will do? It will also change its fluid concentration to 15. Okay. If the external environment further changes from 15 to 10, now the animal is in problem. Now the animal is not going below 15 now. Now this zone is called as the zone of tolerance. Now you can note in your copies, this is the zone of tolerance where the animal is tolerating that particular change. Now what is the change? The external environmental change is 15 to 10 and animal is tolerating it. Still the animal is living and it is tolerating that particular change. Now if the external environment it changes further, if it is going down from 10 to 5, suppose, now what the animal will do? Now the animal will try to leave that particular place. Now this is called as the zone of not tolerance, this is called as the zone of avoidance. Now here the animal will avoid that particular change. So this change is very, very kind of, you can say, negative change for the animal. So if the salt concentration drops from 10 to 5, this is a very high kind of a negative point for the animal, and the animal will avoid this particular change and it will try to leave that place and it will try to move somewhere else where it can have better salt concentrations which are near to 25. So students, if that animal is not able to move out of that place in a particular time, it will die. And this is the third point. Death is the third level after the zone of tolerance, the zone of avoidance, And if avoidance is inevitable, animal will die. So I think you are very much clear about these zones. The first zone is the zone of tolerance. Second zone is zone of avoidance. Third is the death. And the same happens if the salt concentration increases on the other level. If it becomes 30 and then 35 and then 40, the same happens on that side also. So all the animals, they have a particular range of changing their inside environment according to the outside environment. If the animals, they have narrow range, they are called as stenohaline type of animals. And if they have wide ranges, they are called as euryhaline type of animals. So if an animal is a euryhaline type, it will be having broad zone of tolerance and a broad zone of avoidance. And in that particular animal, the death will be like it is far away. In case of stenohaline, the death is quite near, as the zone of tolerance and the zone of avoidance range is quite narrow. Now students, here we have two kinds of animals depending upon the mechanisms of the change of the internal environments. Now, the first type of the animals, these are the conformers. And these conformers, they are changing their internal environment according to the external environment, as we have taken an example in the last slide. So what they do is that they have internal conditions similar to the external conditions. So they are very similarly changing according to the outside conditions. Now what are regulators? They defend a nearly constant internal state. So they are not changing their internal conditions according to the external environment. So suppose if animal is living at 25 millimoles of sodium ions, and if they drop down to 20 or 15, but these animals will be you can say conserving or regulating a 25 millimole sodium ion concentration inside their bodies. So this regulation is due to some kind of regulatory mechanism inside the animal. So that can be some excretory organ, that can be some reduced kind of a permeability, or that can be some active uptake or active outtake of the salts. So as we have discussed in the very you can say start of the lecture, the three mechanisms of regulation. So on the mechanism basis, animals can be conformers, they can be regulators. So you can have a question on these two kinds of animals. Now from the book you can get more of the examples into Osmo Confirmers and regulators. So I can give you few examples. Like if we go with the Hyas, Hyas. So this animal is a crustacean, and what happens that it is poikiloosmotic. What is poikiloosmotic? So they are changing their internal conditions according to the external conditions. So they are conformers basically. So when these osmoconformers, they are placed in dilute sea water. I am saying dilute sea water. Now what will happen? The water will enter the body of these organisms. So that is simple endosmosis. Now the salts, they are lost. So what will happen? If the water is coming inside the body of the animal, the salts are lost. They have got less concentration inside, as we have more of the water inside. So what will happen? That through the excretory organs and the body surface, what will happen? They swell in the dilute sea water. So this is very simple endosmosis phenomenon. If they are put into concentrated seawater, they will shrink. So to some extent, I should say over here that if you take the example of Hyas, it will swell in dilute seawater, it will shrink in concentrated seawater, but they are showing some capacity for the regulation of their internal volumes of the water. So what I mean to say is that if Hyas is uh, kept in the dilute seawater, it will swell, but not to that extent of conformity that it will like burst out. So it can regulate to some extent its volume. So if we draw a curve, the conformer curve will be like this. If you take a graph, so it will be, if you take x-axis like this and y-axis on this side, the line of conformation will be, it will be like this. So what this line will give you, the result is that as we change the internal environments, the external environments, they also change accordingly. So this line will give you the conformity of the animal. Now second example can be of Eplesia. Now this Eplesia, if it is placed in suppose 80% of seawater, first of all it will gain some weight, but after that, after a particular time period, it will enter into a recovery phase. So that swelling will be recovered and it will be coming back to its normal position by losing some of the weight. So that is a regulation kind of a thing. So first of all, Eplesia is showing conformity and then it goes to regulation. So if we draw a curve of this particular animal, what will be the curve? So if you have x-axis on this side and y-axis on this side, that line was the conformity. So the line for Eplesia will be, first of all, it will be conforming, then it will be regulating. So it will come on a plateau. So this kind of a line will be for, first of all, it is conforming, then it is regulating its internal environment. So students, these are some of the examples for conformers and regulators. You can take more of the examples from the book. Now, if we talk about the ranges of the animals to withstand the external environments, so I've told you that these are of two particular types. The first is the narrow range animals, they are the stenohaline animals, and the wide range are called as the euryhaline animals. So sometimes this particular range of euryhalines, these are quite wide ranges. If we take the example of Mytilus, this is a mollusk, and the osmotic pressure of the blood, it is closely following the surrounding water. That means it is conforming, it is a conformer. So it is closely following the surrounding water. If it is living at the salinity of 30% or 15% or even 5%, the blood of Mytilus will go with the surrounding water. So it is showing a high level of conformity. So I am talking about euryhaline ranges. So the range can be from 5% to 30%. So this is a quite high range for the Mytilus. So that is a euryhaline kind of animal. Now, the type of the response of the animal, whether it is conforming or it is regulating, this is also leading to some kind of stress to the animal. So if you are living in an environment which is fresh water and you are under the stress that water is always coming towards the inside. So this is a huge stress. So to cope up with that particular stress, the animals, they have developed their own mechanisms to cope up with the particular problem of the water. Now students, we have got two more, you can say, the divisions of the animals. First is the ionoconformer. So what is ionoconformer? It exerts little control over the solute profile within its extracellular space. Now what is conformity is that changing internal conditions according to the external conditions. So there is very little control over the solute profile. And if we go with the ionic regulators, so they control most of the ions in the extracellular fluids. Now here, the control levels, they are quite high. As we have discussed in the very start of the lecture, that some animals like Eplesia or Astacus or Carcinus, they are having high concentrations of sodium, potassium, but they have got low concentrations of the sulfates. So these are because of this control level. Now they can employ a combination of ion absorption and excretion. So for excretion, they may have some excretory organs, and for ionic absorption, they may have some particular pumps in their general body surface to actively uptake the salts. So this can be one of the mechanism that we have already discussed in the very start of the lecture. So these are few more, you can say, terminologies. This is Osmo Confirmer. So that will be conforming the osmolarity of the internal environment with the external environment. And if the external conditions they change, internal osmolarity changes with it. So that is conformity. And the graph is a linear line. If you go with the osmoregulator, it will maintain its internal osmolarity. So this is the main feature of the regulator. And depending upon the condition, the animal could have an osmolarity higher or lower than the surrounding water. So this is very obvious that if animal is living at 5 millimoles of a particular salt, and if you have the outside concentration at 15, it will be okay at 5. There is no problem. So it is regulating a low level of salt inside its body. So that pertains to a particular animal, that how it wants to live. Now these are some of the terms which are related with the osmotic tolerance. So as we have earlier discussed, we have stenohalines with the narrow range and urehalines with the wide ranges. So no predetermined relationships between strategy and the degree of the tolerance, they have been given out. So we have taken the concept of the tolerance also. We have the concept of avoidance, then is the death. So there is no particular relation of the degree of tolerance with the strategy. So that can be animal's own kind of strategy which it want to emphasize upon in a particular condition. Now students, this is a very, very important graph for you to understand. Now if I give you the very first line, suppose we are taking the first line. This line is this. This is the first line. And this is for a urehaline animal. Now what the line is telling us, this is the internal osmolarity, this is the external osmolarity. So this is the internal medium, this is the external medium. So this, can you give me the answer for this, that this line is for a conformer or a regulator? Yes, this is for a conformer. So I have told you that the line, if it is going like this is for conformation. So as the internal condition, it if suppose if we are taking external condition, so if the external condition is changing from here to here, the internal is changing from here to here. So they are almost same. So that's why the line is like this. So this is for conformation. And this is for a euryhaline animal. Now why it is for euryhaline? Because it has got a wide range of, you can say, for the osmoregulatory change. If it is very small, just in the case of 2, if we are taking number 2, now this particular line is giving you the, now we are concentrating on this particular line for number 2. Now here, what you can give is that it is a conformer, but it is of narrow range. So it is a stenohaline animal, but it is a conformer. This graph is for euryhaline animal, but this is a conformer. But if we go with this particular line, number 3, it is not a conformer, but it is regulating its internal environment. Now, if you see this particular line, number 3, now here we have external change from here to here. This is the external change. But what is the change of internal environment? Suppose from this point, it is starting from near to 500 and it is going only up to 5, 10 or 20. So this is a very less change as compared to a big change in the external environment. So that means this line is for a regulator, and this is a euryhaline type of animal for regulation. And if we take the number 4 curve, this is a smaller curve, this is again for a regulator, but it is a stenohaline of a narrow range kind of animal. So I think that you have understood this particular graph. Now students, what is the necessity of water? So all the animals, they are requiring some source of water. They can be external or they can be internal metabolic water. Now if I am taking the dietary water into account, the diet, what is a diet? It is a mixture of water and solutes in various chemical forms. Now if you take the example of suppose a rat, so it is taking dietary water, it is taking in the food, it is making its own metabolic water. And if you are taking some aquatic animals, they ingest high amounts of liquids, high amounts of water. And if you are taking the plant animal tissues, they are again important source of the dietary water, as all the cells of the plants and the animals, they are containing cytoplasm. So they are a good source of water. And these are some of the, you can say, the sources from which you can take water. What is dietary water? This water is preformed in the food and it is either trapped within the solid food or as a liquid component of the meal. So you may have soluble food, you may have particulate food, you may have solid foods, you may scrap it, you may bore it to ingest, you may chew it, you may swallow it, and you may drink it also. So there are large types of food that you are taking in. So that is all the dietary water. Now the animals, they cannot consume all of the dietary water because some must be retained to give feces the appropriate consistency. So this is very true for the feces and it is very true for the urine also. So the animals, if they are taking in like 100 ml of water, 10 to 20% percent of this water will be given out with the feces, and near to 10 or 20% is given through the urine. So what is left is 60%, and 10% may be like not absorbed or it is going out of the body. So remaining is the 50, and out of this 50 I think 30% is absorbed by the body. So you have to take more and more of water. This is very important. Now once ingested, many macromolecules, they undergo hydrolysis as a part of the digestion. So minor investment of water early in the digestion can take place. Now the later metabolic processes, they generate water. So this is called as the metabolic water. And this water is a very good source for many of the animals. Now this is one of the example of the kangaroo rat. Now this particular rat is making 100% metabolic water, and it is not taking the water from outside. Now this particular slide is giving you the osmoregulation by different kinds of vertebrates. Now if we go by the examples, suppose if you take a lizard or you take an amphibian, first of all, so it is living in strongly you can say hypertonic medium. What is the urine concentration? It is hypotonic. So the skin, it absorbs sodium ions from the water. If you take marine reptiles, they are isotonic, giving out the urine in isotonic form. It drinks seawater, and it has got salt gland which is excreting out the excess of the salts. If you go by the marine birds, so they are giving out the weak hypertonic urine. It drinks seawater, and the salt gland, it secretes excess of the salts. So many birds, they are having the salt glands for the excretion of the salts, and they are present on their beaks or near to the cloaca. So they are excreting excess of the salts, and they are retaining some of the water. So if you go by marine mammals, they are giving out strongly hypertonic urine. So they does not drink any kind of seawater. If we go with the terrestrial bird, it drinks fresh water, and they give out the weakly hypertonic urine, as they have to conserve the water. If we go with the desert mammals, they are strongly hypertonic in urine giving, and they drink no water, and they obtain the water from the metabolic processes, as we have discussed in the previous slide in the case of kangaroo rat. So these are some of the animals which are performing some kind of regulatory mechanisms for osmoregulation. So students, in the next lecture we'll be talking about these solutes which are perturbing solutes or which can be compatible solutes.